today our topic of discussion is glucose. First of all, we talk about the carbohydrates. Carbohydrates are the molecules in which water molecules are combined with carbon molecules. That's why they are known as carb and hydrates. They are known as water carbon. The simplest carbohydrates are monosaccharides, and there are only three monosaccharides: glucose, fructose, and galactose. It means no matter what carbohydrate you ingest, finally they are broken down into only three monosaccharides, which are glucose, fructose, and galactose. Then there are disaccharides. Disaccharides are the molecules in which two monosaccharides are combinedly present. The two important disaccharides that you must remember are lactose and sucrose. Lactose is broken down by lactase into galactose and glucose. So you must remember them as GG. Then sucrose is broken down by sucrase into fructose and glucose. Sucrose is also known as table sugar. So the important MCQs that you must remember here is disaccharides are broken down into monosaccharides into gastrointestinal tract or GI tract. This is important MCQs. And then there are polysaccharides. In polysaccharides, lots of monosaccharides are combined and they form polymers. They are called polysaccharides. There are three important polysaccharides that you must remember. One of them is starch, glycogen, and cellulose. All of these are glucose polymers in which glucose molecules are present and they form polymers. Starch is present in plants, glycogen is present in animals, and cellulose is also present in plant. It is also a glucose polymer, but this has different types of bonds between glucose molecules than starch. The bonds in starch can be digestible in our body, but cellulose are indigestible. So that's why the cellulose has a very clinical importance, that the patients who have constipation, we tell them to take fibers in the diet. These fibers are basically cellulose, and since cellulose cannot be ingested, we cannot digest them, so they add bulk to the stool and it relieves the constipation of the patient. Now we talk about important MCQs that can come into your paper. You must remember, as I told you, that almost all the carbohydrates finally broken down into glucose, fructose, and galactose. So they would ask you a question that the patient has taken rice, and after 30 minutes, what would rise in his blood? So in the rice, the carbohydrate which is present is starch, and as I told you that the starch has glucose polymers present in it, so the level of glucose would rise in the blood. Only monosaccharides can go into the blood. Disaccharides and all of the polysaccharides, they would first broken down into monosaccharides. This is the concept. Secondly, we talk about the glucose entry into the cells. Glucose entry can be done via only two pathways: sodium independent and sodium dependent. Sodium independent entry means that glucose does not need the help of sodium to enter into the cells. It is only done with the help of transporters, and there are 14 type of transporters from GLUT1 to GLUT14. We will talk about them in detail. Just wait a second. Oh, second is passed. Okay, and then we talk about sodium dependent. In sodium dependent entry, we use ATP because it is mostly done at a place where glucose needs to be transported from low concentration to high concentration. That is why we need energy, and these are also called ATP based sodium dependent channels, or you can find different name in which ATP is related to sodium dependent, and it is done at only two places. First of them is intestinal epithelium, and secondly is renal tubules. These MCQs are very important there, and they often come into the exam. They would ask you that the patient, that what type of cells have sodium dependent entry of the glucose, and in options they would give you intestinal epithelium mostly. Now we talk about the glucose metabolism. Glucose can be converted into pyruvate by the process of glycolysis, which we will talk in detail today. And glucose can go into HMP shunt, and glucose can make fatty acids, and glucose can make glycogen via glycogenesis. So all these pathways have detailed lectures. Um, Here one thing I would like to mention, that when glucose is converted into pyruvate via glycolysis, this pyruvate can whether enter into TCA cycle, or it can go into anaerobic metabolism to form lactate. This anaerobic metabolism means when there is decreased oxygen into our body, then pyruvate is converted into lactate, and it is mostly done in the red blood cells. We will talk about this in detail later. Now we talk about different organs and their important correlations with the glucose. First of all, we talk about the brain. Brain needs constant energy, and since it needs constant amount of energy, it requires constant supply of glucose to do the TCA cycle. And for that, the transporter in the brain is called GLUT1. This transporter is insulin independent. It means whether there is insulin or not, the brain transporter picks up the glucose to do the TCA cycle. Secondly, the muscles of heart. Muscles in our body, skeletal muscles and heart muscles. The skeletal muscles mostly use GLUT4, which is insulin dependent. It means they mostly uptake glucose when there is more glucose in our body, and as a result more insulin release. So the transporter which I use is GLUT4. Then we talk about the liver. Liver uses GLUT2. Liver has a special function. It's not only do the glycolysis, it converts glucose into pyruvate and then go into the TCA cycle, but when and it also stores glucose as glycogen via the process called glycogenesis. So it has a double function, and it also does gluconeogenesis. When our body is deprived of glucose and we need glucose, then liver also makes glucose. So GLUT2 is mostly a transporter which is bifunctional. It performs both functions. We will talk about this in detail when we study the gluconeogenesis. Then there is red blood cells. Red blood cells, as I just talked here, they do anaerobic metabolism. They do not have oxygen, and they do not have mitochondria. Since mitochondria binds oxygen with the glucose to do the to produce more energy, since red blood cells have no mitochondria, so it is deprived of oxygen, so only anaerobic metabolism happens into the red blood cells, and it also needs GLUT1 transporter. All you need to remember is, in red blood cells there is no mitochondria, anaerobic metabolism happens, and via glycolysis red blood cells makes lactate. Then comes the adipose tissues. In adipose tissues, GLUT4 transporter is used, and since this transporter is insulin dependent, it means when there is more insulin present in our body, then only this transporter would start transporting glucose into the adipose tissues, and adipose tissues would store that glucose in the form of fatty acids. We will talk about this in detail in fatty acid synthesis. Now I have written some of the important points. You can say as a summary that GLUT1 is present in brain and red blood cells. It is insulin independent. You can see here in brain and red blood cells GLUT1 is present. Secondly, for GLUT2 you must remember it is bidirectional, and it is also used in gluconeogenesis as well as in glycolysis. It is also insulin dependent, and the only insulin dependent transporter is GLUT4, and insulin is required to transport glucose in skeletal muscles and adipose tissues. So this I have mentioned here in muscles and in adipose tissues. And secondly, here do not forget to remember at least these two MCQs, that sodium dependent entry of glucose is done in intestinal epithelium and renal tubules. Cellulose is required to relieve constipation. It is the fiber in diet, and all the carbohydrates are broken down into glucose, fructose or galactose, and only the level of these three would rise in blood no matter what carbohydrate you ingest. If you ingest dairy products, then dairy products include lactose, the galactose and glucose levels would raise in the blood. If you test fruits or table sugar, then the level of fructose and glucose would rise in the blood, as I write here Fg. If you eat rice, rice is rich in starch. Starch has glucose polymers, only glucose levels would rise, and like that. So now we would talk about the glycolysis. Glycolysis is a process in which glucose is converted into pyruvate. This process happens in cytoplasm, and the purpose to do the glycolysis is to make ATP and NADH. So glycolysis is a process in which glucose is converted into pyruvate in the cytoplasm to make ATP and NADH. Now we talk about the stages of the glycolysis. There are 3 stages. The first stage is called the priming stage, the second stage is called the splitting stage, and the third stage is called the energy state. First of all, we talk about the priming stage. In priming stage, first of all, glucose is converted into glucose 6-phosphate. Glucose 6-phosphate is converted into fructose 6-phosphate, and fructose 6-phosphate is converted into fructose 1,6-bisphosphate. In whole process, in this priming stage, there are 2 irreversible steps: glucose to glucose 6-phosphate conversion and fructose 6-phosphate to fructose 1,6-bisphosphate. I have mentioned them with the red pointer. Now we talk about the enzymes. First of all, the enzyme that converts glucose into glucose 6-phosphate is called hexokinase or glucokinase. These are 2 different enzymes and they depend on the type of cell. We talk about them here. First, we talk about the hexokinase. Hexokinase is present in the most tissues. It has no effect from the insulin, and it has low Km and low Vm. I will tell you about these. Most tissues means that hexokinase is present in almost every tissue in our body. It does not depend on insulin, it is insulin independent, and low Km means it requires very low value of glucose to start working. Low Km means that hexokinase is an enzyme which requires very low amount of substrate to start working at the maximum speed, that is Vm. And low Vm means maximum speed at which an enzyme can work. But the maximum speed of hexokinase is very low as compared to other enzymes. So hexokinase it has low Vm and low Km. I hope that you understand this. Now we talk about the glucokinase. Glucokinase is induced by insulin. It is only present in liver and pancreas, and it has a very high Km, and also I would like to tell you that it has a very high Vm. I'll tell you about these. High Km means that it needs a very high amount of substrate to reach its maximum speed, that is Vm. And high Vm means that the working speed of glucokinase is very high. It means it can I will give you an example that let's say if hexokinase Vm is 10, it can convert 10 molecules of glucose in 1 minute, then the speed of the glucokinase would be 100, let's say. It means that it has a very high Vm. And now we compare these two. Hexokinase is insulin independent, glucokinase is insulin dependent. Glucokinase is only present in liver and pancreas. Hexokinase is present in almost every tissue of the body. Hexokinase requires very small amount of glucose to start working. It has low Km and its highest speed is also very low, that is low Vm. And glucokinase has a very high Km, high Vm. It needs very high amount of glucose to start working, and it has very high speed at which it starts working. Now we must understand that glucokinase stops working when there is very small amount of glucose. As I told you before that glucokinase needs very high amount of substrate to start working. It has a very high Km. So when there would be low glucose, it would become inactive. And secondly, it is inhibited, this enzyme is inhibited by fructose 6-phosphate. I will tell you here. Hexokinase is inhibited by glucose 6-phosphate. It means when there would be high amount of glucose 6-phosphate into the cell, hexokinase would be inhibited and stop working. But glucokinase is inhibited by fructose 6-phosphate. When glucose 6-phosphate is converted into fructose 6-phosphate and there is too much level of fructose 6-phosphate, it would automatically inhibit the glucokinase enzyme into the liver and pancreas. Fructose 6-phosphate makes glucokinase inactive. To understand this, we need to know about a protein called glucokinase regulatory protein. What this protein does, it binds with the glucokinase and translocates it into the nucleus in the presence of fructose 6-phosphate. So when glucokinase translocated into the nucleus, it becomes inactive. When glucose comes, this glucose competes with the glucokinase regulatory protein and it translocates the glucokinase back into the cytosol, and when it comes into the cytosol, this glucokinase becomes active. So very high amount of glucose is required to compete with the glucokinase regulatory protein. That is why we say that glucokinase has a very high VM. It only activated in the high amount of glucose. Now we talk about the glucokinase deficiency. It is very important to know about this because glucokinase deficiency would affect only liver and pancreas, as we know. Liver has also many other processes, but pancreas, in pancreas there mild hyperglycemia happens because glucose starts accumulating here and it is not converted into glucose 6-phosphate, so there would be mild hyperglycemia. But the concept that you need to understand here is when there would be low glucose 6-phosphate because of glucokinase deficiency, the machinery of the cell would interpret this information as hypoglycemic state. There would be mild hyperglycemia, but the cells of our body would interpret low glucose 6-phosphate levels as that there is no glucose. That is why there is no glucose 6-phosphate. They would interpret this as hypoglycemia, and as a result there would be low insulin production into our body. This is a very important MCQs. I have seen it in some high yield MCQs that when there is low glucose 6-phosphate, there is low insulin levels. Okay. Now fructose 6-phosphate is converted into fructose 1,6-bisphosphate by an enzyme called phosphofructokinase 1. This is the rate limiting enzyme and this is the rate limiting step of the glycolysis. But how this process whole happens, I will tell you that the fructose 6-phosphate first converts into fructose 2,6-bisphosphate instead of converting into fructose 1,6-bisphosphate. The only difference between this molecule and this molecule is that it is fructose 2,6-bisphosphate. It means that one molecule of phosphate is attached to the second carbon, while here the one molecule of phosphate is attached at the first carbon. First, fructose 6-phosphate is converted into fructose 2,6-bisphosphate by the enzyme called phosphofructokinase 2. Here the enzyme is phosphofructokinase 1. The enzyme that converts fructose 6-phosphate into fructose 1,6-bisphosphate is called phosphofructokinase 1, and that converts into fructose 2,6-bisphosphate is called phosphofructokinase 2. Now this enzyme is bifunctional. This is a very important concept. Bifunctional means when the process of glycolysis happens, then fructose 6-phosphate is converted into fructose 2,6-phosphate by this enzyme, and it is called phosphofructokinase. When the process of gluconeogenesis happens, then this enzyme converts fructose 2,6-phosphate back to fructose 6-phosphate, and then this enzyme is called fructose 1,6-bisphosphatase 2. Alright, so this is the same enzyme actually. This concept is very difficult to understand as it is written in some books. People consider that this is a different enzyme, this is a different enzyme. No, this is a single enzyme, but its name changes according to its function. All right, and then when fructose 2,6-bisphosphatase is made, phosphate is made, it activates the phosphofructokinase 1, the enzyme that converts fructose 6-phosphate into fructose 1,6-bisphosphate. Now listen, this enzyme is made from this enzyme, and after that this enzyme activates this product actually activates this enzyme, which converts the remaining fructose 6 phosphate into fructose 1,6 bisphosphate. पहले fructose 6 phosphate से fructose 2,6 bisphosphate बनता है, फिर fructose 2,6 bisphosphate एक्टिवेट करता है phosphofructokinase 1 को, जो कि बकाया बची हुई fructose 6 phosphate को कन्वर्ट कर देता है fructose 1,6 bisphosphate में. This is how fructose 6 phosphate finally converts into fructose 1,6 bisphosphate. Now, what is the importance of fructose 2,6 bisphosphate? First of all, this is the on-off switch of glycolysis. This is a very important concept. And what is the effect of insulin and glucagon upon the production of fructose 2,6 bisphosphate? Actually, what happens that insulin dephosphorylates the enzyme phosphofructokinase 2. Dephosphorylate means this enzyme has a phosphate group attached to it. Insulin removes that phosphate group, and then this enzyme becomes active and it starts converting fructose 6 phosphate into fructose 2,6 bisphosphate. And when the level of fructose 2,6 bisphosphate rises, the process of glycolysis increases, and when the glucagon comes into our body in the fasting state, the level of insulin decreases, glucagon comes into our body, it adds phosphate group back to phosphofructokinase 2. When a phosphate group is attached to this enzyme, it becomes inactive and the level of fructose 2,6 bisphosphate starts decreasing and it starts converting back to fructose 6 phosphate. So, to make it simple, when fructose 2,6 bisphosphate level increases, glycolysis increases. When the level of fructose 2,6 bisphosphate decreases, glycolysis decreases, gluconeogenesis increases. Now, there are two things that increases the glycolysis. First of all, the level of fructose 2,6 bisphosphate, and secondly, the increased level of AMP into the cell. As I told you before, that the main purpose of doing glycolysis is to produce ATP, is to produce energy. So when there would be more AMP into the cell, it means the cell lacks energy and cell need to produce ATP, then the process of glycolysis increases. I also mentioned this here. What are the key inducers of glycolysis? Fructose 2,6 bisphosphate, which is the on-off switch of glycolysis, and increased level of AMP. And what are the key inhibitors? What are the factors that inhibit the glycolysis? First of all, ATP itself. It is a very common sense that when there is already energy into the cell don't need to produce more energy. And secondly, citrate. Citrate is a molecule that is produced into the TCA cycle. TCA cycle produces energy into the cell. So when there is more citrate into the cell, it means that already energy is being produced, so cell does not need to do glycolysis. So these two steps inhibit glycolysis and these two steps promote glycolysis, these two molecules. That insulin dephosphorylates phosphofructokinase 2 or fructose bisphosphatase 2, then it becomes active and it makes more fructose 2,6 bisphosphate. Here, insulin dephosphorylates phosphofructokinase 2 and it makes more fructose 2,6 bisphosphate, which further activates phosphofructokinase 1 and promotes glycolysis. So insulin comes in the fed state, so we can say that in the fed state the level of fructose 2,6 bisphosphatase increases. In the fed state, insulin comes, level of fructose 2,6 bisphosphatase increases, as a result glycolysis increases. I am repeating these again and again because this is very important concept and it comes in very MCQs. In the same way, when glucagon comes, it adds phosphate group or it phosphorylates phosphofructokinase 2, or you can say fructose bisphosphatase 2. As a result, this enzyme becomes inactive, the level of fructose 2,6 bisphosphatase decreases, and when its level decreases, the glycolysis stops and gluconeogenesis happens. So we can say that glucagon decreases fructose 2,6 bisphosphatase in fasting state. or you can simply remember in fasting state level of fructose 2,6 bisphosphatase decreases. MCQs can come in any form, whether they would ask that in fed state what would happen. You can say in fed state level of fructose 2,6 bisphosphatase, or they would ask in fasting state what would happen, or they would ask in at insulin what would happen, or they would ask at glucagon what would happen. So I tried to summarize all these things here so that it would be easy for you, even if you don't remember the concept, and just utilize this. All right, so this was all the priming stage, and the important concepts are about the priming stage. One thing about glucokinase that I would like to mention here, that I told you that glucokinase becomes inactive when there is decreased level of glucose and there is increased level of fructose 6 phosphate. Now how fructose 6 phosphate affects or inhibits glucokinase? Remember this. There is an enzyme called glucokinase regulatory protein. This is a protein. It binds with glucokinase, and after binding it translocates glucokinase into the nucleus in the presence of fructose 6 phosphate. So when there would be more fructose 6 phosphate, the more glucokinase will enter into the nucleus, and in the nucleus it becomes inactive. When glucose comes into our body, it competes with the glucokinase regulatory protein. It binds with the glucokinase and it takes glucokinase back to the cytosol where it becomes active. So here I have written, in the nucleus glucokinase becomes inactive in the presence of fructose 6 phosphate, and in the cytosol the glucokinase becomes active. Now what will happen when there is glucokinase deficiency? As I told you before, the glucokinase is present only in liver and pancreas. So there would be mild hyperglycemia, very mild, and it would mainly happen in just pancreatic cells. But the important correlation is pregnancy. In pregnant females, there could be gestational diabetes. The pregnant females who have glucokinase deficiency, they might develop the gestational diabetes. And this MCQs often comes that in glucokinase deficiency, here glucokinase deficiency, there would be decreased level of glucose 6 phosphate. When there would be decreased level of glucose 6 phosphate, our cells in the body would consider this a hypoglycemic state, and as a result decreased insulin would be released into our body. So when there would be decreased glucose 6 phosphate, there would be decreased levels of insulin into our body, and there would be mild hyperglycemia in glucokinase deficiency. Now talk about the fructose 2,6 bisphosphate. These are the some important points I have written. This is on off switch I talked about, and it controls the rate limiting step since it activates the phosphofructokinase 1. So guys, this was all about the priming stage. Don't worry, if you just understand this, in the end I would summarize all these concepts. Now we talk about the splitting stage. Splitting stage is very simple. The fructose 1,6 bisphosphate is converted into glyceraldehyde 3 phosphate, and it is converted into two molecules actually. First, fructose 1,6 bisphosphate is converted into another enzyme, and that enzyme is unstable, and that is converted back to glyceraldehyde 3 phosphate as well, but that is not important to remember, so I did not write it here. And in the last, the energy stage comes. Energy state is one molecule of glyceraldehyde 3 phosphate is converted into 1,3 bisphosphoglycerate. 1,3 bisphosphoglycerate is converted into 3 phosphoglycerate. In this conversion, NAD is converted into NADH, and in this conversion one molecule of ATP is released as a source of energy. Then 3 phosphoglycerate in two steps is converted into phosphoenol pyruvate. Even if you forget all of these, just remember that phosphoenol pyruvate is converted into pyruvate via an enzyme called pyruvate kinase, and this is the last irreversible step of glycolysis. So in energy state, 2 ATPs are produced by one glyceraldehyde 3 phosphate. one by the conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate, and one by the conversion of phosphoenol pyruvate to pyruvate. And since we talked that there are two glyceraldehyde 3-phosphate molecules, so as a result, two molecules, each one producing 2 ATPs, so there would be total 4 ATPs produced by one glucose molecule. I have written summarize this. One glyceraldehyde 3-phosphate produces 2 ATPs, one glucose molecule produces 2 G3P, so total 4 ATPs. Now, what happens to this pyruvate? This pyruvate would enter whether into the TCA cycle in the presence of oxygen and mitochondria, where it would produce more ATPs and more energy, and when there is less oxygen or there is no mitochondria, then this pyruvate would enter into the lactate. This mostly happens into the red blood cells, as I mentioned before that red blood cells has no mitochondria, so red blood cells cannot use oxygen, they are deprived of oxygen, so anaerobic metabolism happens in red blood cells, there is no TCA cycle happens in red blood cells, so all they can do is to produce only 2 ATPs, because 2 ATPs are used up in the priming stage when glucose was converted into glucose 6 phosphate, and when fructose 6 phosphate was converted into fructose 1 6 phosphate. I should write here 2 ATPs were used and 4 ATPs were produced. So in anaerobic metabolism, the net profit was 2 ATPs. So red blood cells have only production of 2 ATPs. And in this anaerobic metabolism, pyruvate is converted into lactate by an enzyme called lactate dehydrogenase. So people who have lactate dehydrogenase deficiency, their all pyruvate is whether converted into TCA cycle or their pyruvate is stuck here. Now we talk about the lactic acidosis. Let's say if there is less oxygen into our body, the patient is in hypoxia, then this process would not be able to happen. TCA cycle would not be able to happen, and in decreased supply of oxygen, all the pyruvate would convert into lactate, and there would be increased lactic acid production, because no pyruvate would be going for TCA cycle. As I write, there is very low pyruvate would go to TCA cycle. When there would be more lactic acid production, the cell pH would decrease, there would be decreased bicarbonates. So till here, the important thing to remember is how much ATPs are produced in anaerobic metabolism, 2 ATPs. What is the enzyme? Pyruvate kinase enzyme that is used in the last irreversible step, and how much ATPs are produced by glyceraldehyde 3-phosphate. Now we talk about the pyruvate kinase deficiency. In pyruvate kinase deficiency, red blood cells would be most affected, because as I mentioned that red blood cells have no mitochondria. So when there would be no pyruvate kinase, there would be no anaerobic metabolism, and net profit of ATPs for red blood cells would be 0. There would be loss of ATPs actually, and because of that there would be failure of membrane, and when the membrane would fail, these red blood cells would be phagocytosed in spleen. When spleen would phagocytose so many red blood cells, its size would increase, splenomegaly would happen. So the case would represent for pyruvate kinase deficiency that a newborn baby with extravascular hemolysis because red blood cells are losing their membrane, and splenomegaly. Now there is one thing that I would like to mention here, one more, that in glycolysis 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate, and that is continuous. We have talked about this, but there is a special thing about red blood cells, that in red blood cells some of the 1,3-bisphosphoglycerate is converted into 2,3-bisphosphoglycerate. Only red blood cells do this by an enzyme called PPG mutase, bisphosphoglycerate mutase. Why red blood cells do this? We talk about this. Basically, 2,3-bisphosphoglycerate is used by red blood cells because 2,3-bisphosphoglycerate binds with the hemoglobin, and when it binds with the hemoglobin, it decreases the affinity of hemoglobin for oxygen. So Hemoglobin easily binds with the oxygen at high altitude. So the people who live at high altitudes, the oxygen is already very much low in the air. So 2,3 bisphosphoglycerate is then produced by the red blood cells in those people. It binds with the hemoglobin and helps to deliver the oxygen to all the cells in the body. So guys, now we talk about the energy production in the process of glycolysis. Energy production depends upon the type of cells and oxygen. So as I told that when there would be no oxygen, as here no oxygen, there would be anaerobic metabolism and only 2 ATPs would be produced. So in no oxygen, 2 ATPs would be produced, and when there would be oxygen and mitochondria, then what happens? Mitochondria will bind the oxygen with the glucose, and then it will produce 32 to 30 ATPs plus 6 carbon dioxide molecules and 6 water molecules. So why sometimes 32 and why 30? It depends on the shuttle in the liver and heart. These muscles require more energy, so malate aspartate shuttle is used. Just remember the name of this shuttle. It is used in liver and heart, and this shuttle produces 2 extra ATPs, 32 ATPs, and glycerol 3 phosphate shuttle is used in all muscles of the body, and it produces 30 ATPs. There are 3 irreversible steps in the process of glycolysis: glucose to glucose 6 phosphate conversion, fructose 6 phosphate to fructose 1,6 bisphosphate conversion, and phosphoenol pyruvate to pyruvate conversion. These steps I mentioned with the red pointer. 3 main enzymes are used in glycolysis: hexokinase and glukokinase for first irreversible, phosphofructokinase 1 for fructose 6 to fructose 1,6 bisphosphate, and pyruvate kinase for phosphoenol pyruvate to pyruvate. Now uh, the on off switch for glycolysis is fructose 2,6 bisphosphate. How this happens? Insulin adds a phosphate group to phosphofructokinase 2. As a result, fructose 2,6 phosphate increases, and then it activates phosphofructokinase 1, and the process of glycolysis starts. This is the rate limiting step for glycolysis. Lastly, we talk about the energy production. We talk about some deficiencies. In pyruvate kinase deficiency, the case would present as a newborn baby with extravascular hemolysis and splenomegaly. Why there would be extravascular hemolysis and splenomegaly? You already know this. And 2,3 bisphosphoglycerate has a very important function. At high altitudes, it helps the hemoglobin to deliver oxygen to all cells of the body. This is its important. And energy production, in anaerobic metabolism, when there would be no oxygen and no mitochondria, only 2 ATPs would be produced. When there would be oxygen and mitochondria, energy production would depend on shuttle. When there would be malate aspartate shuttle, as in liver and heart, 32 ATPs would produce. And when there would be glycerol 3 phosphate shuttle, which is present in all cells of the body, there would be 30 ATPs production. Thank you so much.